up where we left off? Yes. There you go. Thank you. Just to refresh your memory, on Thursday we were talking about atom structure. We talked about protons, neutrons, electrons, where each of those structures are found, and some general properties about them. Right? We didn't go into the physics level, but we got into enough level to really understand how they contribute to the formation of these molecules that we're going to study today. After we understood basic atomic structure, then we had to understand, well, what are some of the important bonds necessary to make the different types of molecules. So we went through a list, and we talked about a polar versus a covalent, a polar covalent versus a non-polar covalent bond. And then we left off talking in better detail about ionic bonds as a result of an ion, a cation, and an anion bonding together. And then the final thing that we discussed on Thursday was a hydrogen bond. We kind of mentioned it, got through it really quickly, so I want to pick up there, and then we'll look at the different molecules that are important for biology. And so a hydrogen bond is at least going to have what with it? Maybe it's going to have at least hydrogen. Okay? Hydrogen, and most specifically, a hydrogen that's not neutral. It's going to have a charge on it, a partial charge. And so a hydrogen bond is a result of a hydrogen atom with a covalent bond that allows for that molecule to have a negative charge. So that the hydrogen becomes a little bit more what? The hydrogen gives its shares its electron with something else takes its electron, and something else becomes a little bit more negative. So the hydrogen becomes a little bit more stable and positive. Okay, good. When it's positively charged, it now can be electrically attracted to something else that is negatively charged. So a hydrogen bond is just a weak electrical attraction between hydrogen and something else that has what kind of charge? Opposite, which would be negatively charged. You'll see hydrogen bonds most prominently described with water, but it's not the only place where we'll see hydrogen bonds. Some common examples of hydrogen bonding with atoms that help create or contribute to hydrogen ion bonding is when hydrogen bonds with oxygen, like in water, H2O, and we get that polar molecule, as well as nitrogen. Hydrogen and nitrogen can allow for this partial charge, and then therefore this electrical attractiveness to hydrogen and something else that is negatively charged. To get some examples, for example, water itself is a compound, and it's a covalent bond between oxygen and two hydrogen atoms. So it's a whole molecule. Okay? So within the water molecule, you would not call that a hydrogen bond. That would be a covalent bond making up water. However, as a result of that water molecule, we know that the hydrogen becomes a little bit positive. Okay? That electrical attraction allows for it to form an interaction or a hydrogen bond with something else that is negatively charged. And so the hydrogen bond in this image is illustrated by the dashed lines. Okay? You see they're not physically sharing anything. They're not bound with one another. You see how they're just attracted to one another, but they're not physically conjoined, if you will. Does that make some sense? The little dashed lines is just representing like that electrical attraction. We used the example of magnets the other day. Imagine those being two separate magnets. Okay, the hydrogen bond is that electrical attraction in that analogy. The magnets would be water and ammonia. that they would be connected. In addition to, for example, finding it on water, we will find, hydrogen bonds critical for holding the structure of this molecule called DNA, which you've probably heard of. Maybe you don't really understand yet, but you will. And proteins, critical to protein structure as well. I feel confident that I may have mentioned this the other day, but we were kind of rushing through at the end of class. I don't want you to translate weak with not useful. Okay? It's still a bond. It still gives structure to things like water, in a pond or ice or whatever, or DNA or proteins or whatever. So just because it's described as weak, it still has its purpose. It's very useful. What questions do you have about the hydrogen bond before we start looking at our different macromolecules and then also water? Yes?
So the hydrogen bond only pertains to half attraction where another bond,
Like a covalent bond?
covalent bond is with respect to the whole side.
You could use that analogy, yes. If we go back to that image previously, okay, the red and the gray is a single molecule of water. The oxygen and the hydrogen atoms are polar, covalently bound together. So they are sharing electrons. Conversely, water and ammonia, they aren't sharing anything. They're just electrically attracted to one another.
Okay.
But what do you think is making up ammonia then? What type of bond? They would actually be, this thing looks a whole heck of a lot like what? It looks a lot like the water bond, so it's probably what kind of bond? Polar and covalent. Can you see how in this one molecule, part of it is negatively charged and part of it is positively charged? That would also be an example of ammonia, an example of a polar covalent bond. Before I move on, Morgan, did I answer your question?
Yes.
Good. Thank you. So we talked about hydrogen bonds. We will see them again when we look at DNA and when we look at proteins. How about statements on the top of your page 5 of your chapter 2 handout? And y'all get the little text message reminder? Bring your notes? Okay, let's make sure it's working if you signed up for it. So bring your handouts if you're going to choose to use them. Looking at the top of page 5, there's a statement. If you take any other class with me, I'll say this statement at least once a day. And you might want to just commit this concept to application all semester in this class. The structure of something is going to dictate its what? Function. How something's built indicates what it's going to do. You can take that all the way down to the atomic level, protons, electrons, neutrons, up to the molecular level, proteins, carbohydrates, etc., up to an entire organ, say your heart and your body, up to an entire organism, a plant in the field or something like that. The structure in the scientific world, the structure of something dictates its function. We'll get that to almost be like a church response. Structure dictates. I'm just saying it over and over. And so I want you to keep that in mind when we start looking at these different molecules. A couple of images, examples that I have include proteins associated with the cell membrane that serve as what's called receptors, or the structure of your DNA. Okay, and I could just keep going on with the things that we're going to study in just a minute. Before you get on to the different molecules, we can do some practice. Here we have a molecule. It's called methane. You've probably heard of it. Okay. You've heard of methane gas, maybe? It's a compound between which atoms? Hydrogen and carbon. Okay. Molecularly speaking, we could write that as CH, and what would be the subscript? 4. All right, there's 4 hydrogen atoms. But I want want you to look at is the lines between the carbon and the hydrogen atom. Each of those lines represents a covalent bond. So how many bonds are in this methane? 4. You would say 4 covalent bonds make up this molecule. We can do another example of carbon dioxide. Carbon, di means what? 2. And what kind of bonds are these? It's a double, and you could say covalent bond. You can see how it's represented by how many lines? There's 2 double covalent bonds. And they're, to make sense, kind of the illustration. We won't spend so much time on the structure as the what? Good job. We're going to first talk about this molecule in our environment, or this compound in our environment called water. We take it for granted. It's everywhere. And because it is everywhere in our environment, it's within cells, it's around cells, and it supports all the chemical reactions that happen inside of a cell. So we really need to understand the properties of water. One property of water is that it's considered to be cohesive. Anybody read ahead and know what that meant? Yes? Correct. It means they stick together. If a molecule is cohesive, that means it bonds with other molecules of the same type. So in other words, water can form what kind of bonds with other water molecules? Hydrogen bonds. Okay. So what it means if water is cohesive is that water can form hydrogen bonds with other water molecules. The follow-up then is, can water only form hydrogen bonds with only water? No. It could certainly dissolve. You list any substance in your body, probably most are going to be water soluble. And so instead of saying that water is cohesive, it's also what? Adhesive. And how should we define the adhesive property of water? Forms hydrogen bonds with other substances. If it's adhesive. Oh. Sorry, getting ahead of myself. Here's an image representing cohesion. And you can see the dotted lines represent what? The electrical attraction. We call those hydrogen bonds. Okay. So water is definitely cohesive. It's also adhesive, and we said that is when water forms hydrogen bonds with something besides water. The example in your textbook is how water actually gets transported up the vasculature of a plant. It's got to kind of defy gravity, if you will. And so what it does is utilize hydrogen bonding to kind of walk along upwards into those upper structures. So those structures are not water molecules. They're phospholipids and carbohydrates and all kinds of other molecules in there. Are you okay on the difference between cohesive versus adhesiveness of water? Any questions before we move on? Yes.
When you say chemical, right? Is another word for that element?
It could be, or it could be compounds. The word chemical is just pretty vague because there are so many different molecules. I could have listed an element like calcium or a calcium ion or whatever. So in short, yes. It's just kind of a generic way to say something else without having to list everything in the world. Additional questions? I do want to go back to an image that I kind of quickly, anybody see the image that I quickly went over? So I was talking about adhesion. What image did you guys see that was maybe interesting? The what? The bug, right? Let's go back to your cohesive description real quick. Because water molecules are cohesive and they form hydrogen bonds with each other, it contributes to this property of water called surface tension. So in your cohesive notes, I want you to go back and add, it contributes to surface tension. Surface tension. Because water is cohesive, that contributes to water's surface tension, which you can't hardly get a better description of surface tension than this image right here. What can you take away about surface tension? Of the water. Or maybe what can we take away about hydrogen bonds? I said they're weak, right? But look, an animal can walk on water. Although we describe hydrogen bonds as being weak, as I mentioned, they're still functional, they're still efficient. Okay? And they're strong enough to support an animal that's able to distribute its weight equally across the surface. Okay? And so, because water is cohesive, we can see lakes looking flat. If you see some, you pour your milk out of a jug, it looks all cohesive, right? It's because of the chemical attractiveness of the hydrogen bonds of the water molecules. So, water is cohesive and water is adhesive, as we described. In addition, water is considered to be a good solvent. The terms that we're going to describe in just a moment can be very easily confused, so I'll walk through them, present them to you, and then we'll go back and analyze them. Okay? Water is a good solvent. What do you think that means? Breaks substances down. Another way, it's basically restating what we said earlier. Water is adhesive. It bonds with other molecules. And because it's adhesive, it can be a good solvent. Well, when we look at this interaction, when water dissolves something else, we've actually created a solution. Okay? So what's the textbook definition of a solution? One or more solutes dissolved in a liquid solvent. So a solution is a mixture of something called solute and solvent. A solution is a mixture of a solute and a solvent. Biologically speaking, though, our solvent is what? Water. So we're looking at biology, we're going to study water. But maybe you've ever used that goop be gone or whatever. You ever use something like that? That would be an example of a solvent, and whatever you're trying to get rid of would be the solute. Okay? Another example. So what we can do is then, right, so the solution is just a mixture. To understand it better, we need to know what the solute is. So what is the solute? The substance that goes in, that is being dissolved. The solute is a substance that's being dissolved. What is the next thing that we need to define? The solvent. And how do you want to define the solvent? What does the dissolving? Which dissolve? It dissolves the solute. I used the word substances, but I could have just as well used what? I said, and I've got a big fat typo too. The solvent is the substance that dissolves the, that's just a really bad solute. Solute. So right here, I got a typo. And that's just all kinds of wrong right now. We're going to
Yours
use,
is making us feel comfortable. I know that.
did it just for you guys. I need at least one a week, so. So we're going to describe a solvent as a substance that dissolves the solute. Okay, very good. Just technical terms, okay, that we'll put in practice throughout the semester. We can give an example of salt water. This one's easy enough. What is the solvent? Water. The solute? Salt. Okay. The solution? Salt water. Okay on those definitions of a solution? Any questions? Not everything can be dissolved in water. Not everything can form hydrogen bonds with water. However, those substances that can, you can categorically call them hydrophilic. Hydrophilic. Hydro, the prefix hydro means water. Philic means loving. Hydrophilic substances are called water-loving, or more technically you could say water-soluble. In other words, they can form what with water? Hydrogen bonds. Additional clarification that you might want to add in your notes about hydrophilic substances is that because they can form hydrogen bonds, it's due to they've probably got a what? They're probably and what about them would make them dissolvable? Yes. They're polar. Okay. Hydrophilic substances are either polar, okay, so either have a partial charge or are fully charged. So in summary, basically polar molecules or your ions that are fully charged, your cations and your anions. So polar molecules and ions tend to be hydrophilic. Are all molecules polar or charged? No. There's definitely non-polar particles, so do you think they would dissolve in water? Would they form hydrogen bonds? No. And we would call those substances what in water? They would be hydrophobic. Okay? Hydro again refers to water. The phobic means fearing. Water does not have emotions. However, it just talks about how hydrophobic substances will not form hydrogen bonds, will not interact, and therefore they are not soluble. They are not water soluble. Another typo on the note. Water, a good solid, sorry. Getting in a hurry right now. So if I use the word something being hydrophilic, water-loving, water-soluble, has a what? It's polar or an ion. It's got a charge, whether it's full or partial. Hydrophobic substances, spirit, water insoluble. Here's just an animation that I found a few years ago to kind of illustrate when a hydrophobic substance is actually placed in the watery environment. Which molecule up here would be representing the hydrophobic substance? What color is it? the purple-blue thing, okay? And then the water molecules are the red-yellow substances. Okay? You'll see this most when you see oils and waters interacting. You can see how the oil would be considered to be hydrophobic, which is the image on the right kind of illustrating the watery versus the oily environment. Additionally, water is an important buffer for many reasons, and including temperature. What does the word buffer mean? Kind of softens the impact of something. Okay? So water is a temperature buffer. It softens the impacts of temperature on organisms and even just our entire world, the biosphere. Okay? This is where I didn't get to update my notes. So what it does is help regulate heat. In the world, in your body, wherever water is, it's going to regulate the heat of the environment because it can absorb and transfer heat pretty easily. An example is your blood. What happens when you exercise? Your muscles get warm, your blood goes over your muscles, and then your blood goes where? Where can you presume it's going when I can see someone who just exercised, right? What about them might I know that they just exercised? There's two things I can try to see. They're going to be red because the blood is at the surface, and they may also sweat. Okay? So let's talk about blood. What's a primary component of blood? Water. Okay? It's picking up heat and transferring it, in that case, to the surface of the skin to let the heat out. Yes, to try to cool. Okay. Additionally, when one sweats, we have a second way of cooling because now we have water on top of the skin, and what blows over that wet environment? Air, and picking up more. What's going out? Well, besides the water, what's the water transferring? Heat. Okay, it's evaporating, trying to transfer out heat. Okay, so we can see an example like in the body. Well, we can see an example in the body, but we can certainly see how this works in, say, a pond or the oceans, okay, that we have in our atmosphere. Okay. To give further illustration of how how water can buffer heat. Anybody been to a coast or the ocean? Okay. You may be right on the coast, and the ocean wind blows across the ocean, and it picks up the transfer of temperature out of the ocean. And so the wind feels really cool. And then you might go inland, say 7 miles, and it's profoundly hot, okay, because there's not that water in the environment. Okay, so if you go on vacation to a lake or something, you'll notice that the aquatic environment is a little bit cooler than when there is no water, right? So heat allows for a buffer for not just living systems, but also just where there is water in the environment. That's another great example how water can transfer heat is when the water is evaporating in the atmosphere, it's trying to liberate heat, okay? And so it just gets on your body. Instead of getting out of your body, it's clinging to your body, so you get hot and soppy. But if we look at the structure of water, it's interesting, kind of unique. Most substances are most densely packed in their solid state, intermediately dense in their liquid, and then least dense in their gaseous environment. But look at water. What state is water where the molecules most densely packed at? The liquid state. Kind of reversed from most other substances. Water is most solid in the liquid state. Intermediate. Did I just misspeak? Water is more solid in the. This is where we end. It is more densely packed in the liquid. In the solid state, it's intermediate. So usually that's flipped. Substances in their solid state are usually more densely packed. And then less densely packed in the gaseous state. So liquid water, we have an example, right? In the cup. Solid, we have some examples like on my car, like what? Ice, okay? And then sometimes we have an example of water in the gaseous state, when we talked about humidification, sweating, when it goes out in the environment. You can't really see it necessarily. So that's kind of interesting how the density varies. It's backwards from most other states. Water is most dense in the which state? Liquid, intermediate in the solid, and less in the gaseous state. Makes more sense if you look at it with ice. Okay? Ice is solid. If it were more dense in the liquid state, ice would collect where? At the bottom. Okay, would pack. But ice actually floats, which reinforces the fact that water is actually more densely packed with water molecules than the solid or the gaseous state. Okay, we certainly can't see it in the atmosphere with the eye. An important feature of that is if ice does form, it provides insulation for that environment. So think about the ponds, like here in Oklahoma, we think about ponds accumulating an ice layer. Well, all of the animals living in the watery environment, will they automatically freeze? No, because we said water is a temperature buffer. It'll be warmer at the bottom of that pond or whatever than at the surface of that environment. Does that make some sense? Alright, so water is a pretty cool molecule, how it interacts in the environment. Okay? It's very abundant, most abundant molecule. Your book goes ahead and introduces a couple of terms, so I'll introduce them now, but we'll study them in better detail when we get into, I think, our second unit, looking at enzymes and all kinds of stuff. But to understand how molecules are formed, we really need to use these terms. And so we're getting into applying some of our chemical knowledge. And if we look at the formation of any molecule, we could call it a chemical reaction. A chemical reaction is just going to produce something. In order to make something, we need to have what's called inputs. And in a chemical reaction, the inputs are called reactants. The reactants are the beginning molecules in a particular reaction. So they're going to start. Another word you could use is inputs. So they're going to initiate some chemical reaction. What do you think is formed? The output and the product. Okay? The product is what is formed. Okay? Those are the term we could use as output. Product. I think product is a term to use. In a chemical reaction, the reactants initiate something, and then the products are what is the result. Okay. So, for example, when you eat, I'm going to go with mine right here. My coffee, not just coffee. I've got lots of fats and sugars in here, right? Okay. So the inputs would be fats and sugars. When I take them into my body, the output, the chemical reactions will, in my body, because I don't exercise except for when I run up here really fast, chemical reactions would convert them to other forms of fats stored in the body. Okay? So the inputs would have been sugar, the output, the reactant, or excuse me, the products would be, sadly, fat. Okay? Does that make sense? We start with what you had. How they happen is very technical, so we'll look at some chemical reactions coming up this semester. Additionally, when we understand chemical reactions occurring in aquatic environments, we have to take into consideration the pH of the environment. And if you look at your handout, I've got a note about the pH. What does the pH represent? What does it measure? Concentration of hydrogen ions. Circle that. That's the critical part. The pH measures the hydrogen ion concentration of a solution. The pH measures the hydrogen ion concentration of a solution. The pH scale is it's often confused. Right? It's trying to usually be simplified, but sometimes things are too simple and they get confusing. What it does is it tells us whether something is acidic or basic. The word quantitative just gives a value. It's just going to give us the value of the amount of hydrogen ions in a solution. If we look at a scale, and I've got the pH range, and if you look across the board, it goes across your handout, an acid, neutral, or base. Okay, can you with me with your handout? Look, go acid, neutral, base. What is the range of the pH scale? What's the low end value? 0. Okay, so under where it says acid, the pH range is 0. Up to neutral, which neutral is 7, and then basic upwards of 14, the value. Here's where we get confused. This is what's called a logarithmic scale. So on the value of 0 on the acidic end, if we just see this for the first time, we must presume that means no hydrogen ions. But it's actually the reverse. So acidic environments actually have what? High levels of hydrogen ions. So can you see my shorthand notes? Acids have elevated hydrogen ions. You might want to clarify that are free in solution. Acids have a high concentration of free hydrogen ions in solution. Everybody catch that, or do I need to repeat that? Acids have a high concentration of free hydrogen ions. Acids have a high concentration of free. Be okay if they use the word free? They're not bound to anything. Hydrogen ions in solution. Hydrogen ions. The hydrogen ions are free. They're dissolved. Hydrogen ions. So if something has, say, a pH of 2, I would say that substance is very what? Acidic, and it has a whole bunch of what in it? Free hydrogen ions. Not just the hydrogen ions are there and they are not bound, they are free, they're dissolved. Okay? They're able to react. By the flip side, so let's go all the way to the other side. Solutions that are basic or alkaline, probably what? Low or maybe even no levels of free hydrogen ions. I'll just clarify that it's a low concentration of free hydrogen ions. They may be present, they're just bound to something else. So bases have what? Low levels of free hydrogen ions. Hydrogen ions have, if there's a bunch of them in solution, solution is probably acidic. If there aren't many free hydrogen ions, the solution is probably basic, or you can use the word alkaline. Now what about neutral? Will it have free hydrogen ions? It will have some, but they'll be balanced with other molecules that could bind them. Okay, so it would have a, for a simple view, you could say a balanced ratio of free hydrogen ions. Yes, so it's balanced. Okay. So if there's a free hydrogen ion, there's going to be something that it could bind to. But it's balanced so that overall there's not more or less. In our living systems, most environments probably prefer what? Most reactions might prefer what type of environment? Most require a neutral, but not all by any means. Classic examples in your stomach probably requires acidic environment. And just start breaking down molecules. Let's go back to that image that I showed a moment ago. You can understand this. Battery acids, what do you know? Still a battery on you?
Your clothes.
Change your clothes, because what's going to happen to them, or your skin? It's going to dissolve, right? It's going to break them down. Okay? Lemon juice is very acidic. You probably find citrus as an important cleaning molecule because it can break down substances. Coffee, right, without anything added into it, is on the acidic end. Blood is neutral, but it's actually shoving towards the alkaline end. It's like 7.35 to 7.45. Seawater, a little bit above neutral, so on the low end of the basic range. Lye, same thing. You might have used lye before. Heard of it? Like lye soap to break down. Okay, maybe you put lye, porta potty, break down, right? So it is also not something that you would want to get directly on your skin, okay? Because your skin probably would like what kind of environment? Neutral. Pure water without anything else in it, it's been deionized and all that stuff is going to have what pH? Neutral. Okay. When you start adding substances in it, that's when the pH changes. Alright, so if we look at some features about hydrogen ions, acids and bases, we said that acidic environments have more free hydrogen ions because what an acid does is release hydrogen ions. It releases them when placed in the water, which makes for free hydrogen ions. What do you think bases do? Bases must then gather and bind. The term is called accept hydrogen ions when placed in water. An example I have here in the brown is hydrochloric acid. Hydrochloric acid, when placed in water, it releases, can you see the free hydrogen ion? Can you see the reaction? HCl would be mine. And then the reactants, and then hydrogen and chloride would be my reactant is hydrogen chloride, and hydrogen chloride ion would be the product. Okay. Conversely, we said bases do what with hydrogen ions? They actually accept. For example, this one's called sodium hydroxide. When placed in water, we have sodium ion. Notice that there's still hydrogen ion, but it's not what I've been using the word free. It's found in what? Oxygen. So it's there, it's just not free. And so this would be a base. It binds the hydrogen ion from placed in water. Are you with me so far? Acid, low on the pH scale, releases hydrogen ions. Bases, upper end of this pH scale, bind hydrogen ions, neutral or balanced. In biological systems, though, rapid changes in pH are not desirable. It would be lethal to a cell. And so the body, like living organisms, have what's called buffer systems, and they help avoid any fluctuation in pH. They help resist fluctuation in pH. Some organs in your body that allow for this are your kidneys and your blood have built-in buffers that make sure your blood pH is always about that 7.35 to 7.45 range. Because if blood is neutral at 7.45, and let's say my blood pH gets to 7.5, my blood is actually becoming what? Basic, okay, which, or if it becomes, say, 7.3, the blood is then becoming acidic. It changes the entire environment. It may change the structure of things, and it may change the function of things, right? It makes a lethal environment, okay? So whole systems have buffering systems. Our environment has buffering systems. So ocean, how everything interreacts is trying to make sure, right, pH is pretty stable and not crazy fluctuating. And that transitions us to the molecules that I had you guys do your handout on last week. Categorically, the molecules that we're going to conclude our discussion with today are called organic. And I have it up here, but what's an important element that must be present for something to be considered organic? Must have carbon and also hydrogen. The key is the carbon. So in all of these molecules that we're going to study, we're going to see carbon, definitely going to see hydrogen, but then a mixture of other elements to make up these unique macromolecules. The organic compounds that we're going to study and you did your handout over include your carbohydrates, proteins, nucleic acids, and lipids. So at this point, you might want to get your little handout by you. You submitted it. If you want it, I can hand it back to you, but otherwise I don't have it out. So the primary thing with our organic molecules is they're going to contain carbon and hydrogen. Okay. Of all of these molecules, I hope that when you went through and read ahead, you saw a theme through the synthesis of these molecules. Okay. Through 3 of the 4, there was a theme that we saw for making carbohydrates, proteins, nucleic acids, and lipids. What maybe you saw was the use of this term called monomer and polymer. Okay? And this is where I don't have time for this really. I don't have to worry about scribbling. I wish you guys could follow along like what we've been doing. A monomer, mono means what? One. A polymer means many or multiple. Okay? In each of these molecules, to make something complex, we have to start with the building unit. Okay? To make a polymer, we have to start with a monomer. Okay? So hopefully you saw that monomer-polymer relationship when you're doing your handout. Okay? In order to create a monomer, that was a new word. In order to use monomers to create polymers, we can use two primary reactions, including dehydration and hydrolysis reactions. Okay, let's first talk about the dehydration reaction. Dehydration, you could, not even being a chemist, you know what's going on there. Dehydration is removing water. Okay? During dehydration reactions, two monomers are joined together, and when they're joined together, what's a byproduct? What would be a product? Water. Okay? And then also the polymer. Okay? So water is going to be a byproduct. Looking at your handout, what other word might I say besides dehydration? I might say condensation, because you can visually appreciate condensation liberates water. Okay? Water, which could evaporate. Okay. So chemical reactions that join two monomers, when they're joined, if water is liberated, we would call this a word. If I'm adding two monomers together and water is liberated, I would call this a. The bond would be formed, but if water is liberated, it's called a. So I'm taking two monomer subunits, I'm joining them together, and then when I join them together, water's a byproduct. We just said that's a condensation or a dehydration. Okay, I'm just restating it to try to not confuse you just for driving home. So, if two subunits are joined together by some bond, whichever we're going to identify in just a minute, and water is liberated, and we're building something that's called a dehydration or condensation reaction. So if I'm building something big, the individual reactions are called, one more time, condensation, or I could say dehydration. Okay, with me? Okay, if I'm building something, okay, dehydration. Our cells don't just build, they can also break down. Okay? If water is used in that reaction, it sounds similar, okay, but it's called a hydrolysis. Now let's focus on the end of this. Hydrolysis. Have you ever lysed something? What's that mean? To what? Lyse actually means to split, break. Okay? In a hydrolysis reaction, what could be broken? A bond, okay, is broken. Okay? And in order to do so, we actually have to put water back in to the reaction. So if I'm building a molecule, it might be what type of reaction? A dehydration or condensation. But if I break it down, it might be a hydrolysis. Okay, these aren't the only types of reactions to build or break down, but they're two important ones that we'll see for monomer production. So dehydration does what to it? Builds. It creates polymers. Hydrolysis breaks them to release water and, or, let me say that, uses water to release. If I have a polymer and I'm breaking a bond, I'm releasing a monomer. A picture will probably help you a ton with this. Okay? Here's an example. And we're using the word monomer, polymer being super generic. I could use glucose and sucrose. Those would be monomers of sugars. If I want to join them together, what type of reaction? If I want to join this Dehydration. And when that happens, can you see the new bond that is formed? And now the individual monomers are now bound together, linked together, and we're forming a what? Polymer. We're forming a polymer. Can you see how those two monomers that were separate before, they're joined now? Can you see the bond here? Can you see where water is actually taken off in the middle? Can you see how water is released, right, condensed? How those molecules have been dehydrated? A couple of yeses. Any more? The two monomers are separate. The condensation reaction joins them, and as a result, water is liberated. What if I want to break that bond? What kind of reaction could I use? Hydrolysis. Okay. So it's the reverse. Hydrolysis reaction actually splits what have been previously a polymer into individual what? Monomers. So in this case, you see how we're breaking down? You see how the hydrolysis broke something big into smaller subunits? There is some universal, some people on, some people not. So before I move on, I want to double check, triple check. If I'm building a polymer, joining monomer subunits, what type of reaction? Dehydration. Okay. Conversely, if I want to break those bonds and release those monomers, I would have a hydrolysis. Pictorially, right, these images are really oversimplified of the different macromolecules. We're going to talk about carbohydrates, proteins, nucleic acids, and lipids. I've got a short list there on page 7 of your handout. What are carbohydrates? Basically big polymers of what? Sugars. Carbohydrates are polymers of sugars. Proteins are polymers of amino acids. Very good. Okay, so we said carbohydrates are polymers of sugars. Proteins are polymers of amino acids. And what about nucleic acids? They are polymers of nucleotides. Okay. Lipids. What about lipids? That'd be an example. What else? Triglycerides are an example. Waxes and other lipids. Okay. A note I want you to make about your lipids is that they don't fit into this neat monomer polymer. Easy bits. Okay. That's not real technical sounding, but they're not built by the same type of monomers. Between the first three, between our carbohydrates, our proteins, and our nucleic acids, I have a note on there about commonalities. Those three things, in order to build them, we all have to have a monomer to build that respective polymer. They can be built using dehydration reactions or broken down utilizing hydrolysis reactions. We also have to kind of take a side note and talk about that important atom called carbon. Okay, so before we move on to the macromolecules themselves, let's look at carbon. And a little better detail. How many electrons does carbon have? Or I guess say protons or neutrons. Looking at this image. How many total? 6. Okay, good. Sorry. I have 2 in the first shell, and then how many in the second shell? 4. Is it stable? When would it be most satisfied? When the second shell is full. So how many how many electrons could carbon accept in order to make it fully stable? How many is in its shell? Outer shell? 4. How many could that shell maximally hold? 8. So it could accept up to 4 more. Carbon has a valence of 4. And if you remember our example of methane, remember it was CH4? 1, 2, 3, 4. Or the double bonds with carbon dioxide. Okay, it has what's called a valence of 4. And so it has 4 outer electrons, and therefore can very easily form covalent bonds with up to 4 different atoms. And so it's versatile, in other words. We're going to see carbon throughout the semester. We only have about 7 minutes left, so what I'd like to do is have you turn to page 8 of your handout for chapter 2. And I'm going to walk through that little table looking at the different macromolecules. Okay? Just list them in order: carbohydrates, proteins, nucleic acids, and then lipids at the bottom. In the headers, right on the far left column is the macromolecule. The second header is an example of a monomer. Might want to clarify that that's an example. Those aren't the only monomers. And then the third column is an example of a polymer structure. And then the fourth column is what you're going to just give some, to name the bonds. So that's what I'm going to give you. Your book doesn't give you, it gives you some, but not all of the bonds. So I want to make sure that you have those. So let's work left to right. Okay, are y'all with me on page 8 in the handout? Okay. Carbohydrates. What is the monomer subunit of a carbohydrate? We would call that a monosaccharide. Monosaccharide just means one in sugar, basically. Monosaccharide. So in carbohydrates, the monomers are called monosaccharide. An example of a monosaccharide is called glucose. You've probably heard of that one if you've heard like blood sugar, you've got diabetes or something like that. People are referring to this molecule called glucose. Notice that the structure of the sugars kind of have a ring-like structure. You'll see similar ring-like structures in other molecules. So when we have better time, we'll characterize those in just a minute. Individual monosaccharides could join to form a first, if I have 2 monosaccharides, that would first form a disaccharide, then I could add another and call it a, or you could even keep going up, but yes, eventually we would form a tri and so on. And it could form a polysaccharide. So individual monosaccharides can form individual sugars can form a big complex sugar. This would be the polymer generic name. I could see have some joined together. This bond right here is called a bond is called a glycosidic bond that joins individual monosaccharides. This sugar is bound to this sugar via a glycosidic bond. The type of covalent bond between sugars. Real quick, proteins. We'll come back to it, but the bond between proteins, what you can add on that table between proteins is called a, I think it gives you guys this one. It is a type of covalent bond. It's between amino acids, and the bond between those are on the far right column under proteins. The bond is called a peptide bond. So a peptide bond helps form our what? Protein. Glycosidic bond helps form our carbohydrates, our polysaccharides. Go down to the next row where it says nucleic acid. This is not given in your book either, but I want you guys to use it. It's all right on the board. It's called a phosphodiester. So it's a mouthful. That's why they don't give it to you, I guess. Phosphodiester is the name of the bond between nucleotides that make up our nucleic acid. That forms our nucleic acids, like DNA and RNA. We're out of time. We did not finish chapter 2 as I anticipated. So what I'm going to ask you to do is review the polymers. You guys, it sounds like you did go ahead and get in there, and that handout helps, so that I can zip through the macromolecules on Thursday, but we'll also get through organelle structure, because next Tuesday is what? Exam number 1. So we have got to get through chapter 3 on Thursday. Be safe out there, and we'll see you later.